Hello? Yes. Good morning, everyone. Welcome to our microbiology session today. Uh, do we have lights here? let me find lights. Yeah, that's no. Have no idea where is. Oh, he is fixing. He's finding light for us. Oh. Let's first fix the light. Otherwise, being in the dark is not ideal, especially online. I think they cannot see us properly. Uh, but we have time because one of our speakers could not make it today for the session, so we have one slot more. That's 5 minutes, and then I think we go. No light. From the. Oh, okay. It's a bit weird because online they cannot see it. Sweet. Now you want me to tell the order? First is this one. Okay. The second is this. Then the last one is the same person, but she's going to present this and then this. Okay. So 1, 2, 3, 4. Okay. Yeah. Yeah, because it was a change on the. Yeah. Morning. Good morning. Yes. There is light now. Not there. Yeah. That's better. Huh? I think that's better. Yeah. All right. We have lights finally. So welcome everyone. We had a little change. We, as I said, there was one speaker less, and but we have a discussion of microbiology, and we keep our questions for the end, and we have a panel at the end. I would like to introduce our first speaker. She's from the LR, and she's going to talk about biofilms and the successful missions that we had. Like, floor is yours, Katarina.
Thank you. I have to decide which side. Maybe I will stand on this side. I have the point. Okay, thank you very much for having me here. I'm going to present the biofilms experiment here today, which is an experiment about antimicrobial surfaces against bacterial attachment and then biofilm formation under reduced gravity conditions. So first of all, why do we even have to battle biofilm formation in spaceflight conditions? Biofilms can cause severe damage to equipment. So biofilms are these accumulations of microorganisms that can either occur on surfaces or also on liquid air interfaces, are accumulations of microorganisms that form a matrix out of extracellular polymeric substances that can be nucleic acids, lipids, proteins. And this matrix is very slimy here, as you can see here. So biofilms can damage equipment. So these 3 pictures are pictures from equipment of the International Space Station. So here, these 2 pictures are biofilms that have formed inside the water recycling system of the ISS, where you can see here in this picture that every opening here is blocked by a biofilm except this one. And here also this inlet is completely blocked by a biofilm. And then also biofilms can occur as mold, so as the growth of filamentous fungi on surfaces. So this is where astronauts used to hang their wet towels after working out, and then mold had formed just as it does in our bathrooms at home. So yeah, biofilms can damage equipment. Biofilms can also be a threat to crew health though. So biofilms can act as a reservoir for pathogenic species, and then also can act as a source of the spread of these pathogenic species. And astronauts, they have, as we know, a reduced immune function under spaceflight conditions. So there's a question here if biofilms as a reservoir for pathogenic species, it would not be an ideal setup. And then I put a question mark here because there is the question of what the influence of space conditions on biofilm formation actually are. There are some studies that show that biofilms are thicker under microgravity, that they have a changed morphology. But there are also studies that show the opposite, so that biofilms are less robust under microgravity. So there I put here this big question mark. There's a lot of unknown things about biofilms under reduced gravity conditions. One possible solution, I said in advance as a disclaimer, it's not the optimal solution, and I also would say it's not a standalone solution, but it's a mitigation strategy. It's to use antimicrobial surfaces, for example, antimicrobial active metals such as copper or silver. And these metals then prevent the attachment of organisms to a surface, and then so the first step of biofilm formation is the attachment, and from that first initial attachment the biofilms then further grow, and by preventing this first initial attachment then ultimately biofilm formation should be inhibited. We are working with copper as an antimicrobial metal. And copper is known as an antimicrobial agent since ancient times. And it works like this, that when copper enters the cells, reactive oxygen species are produced, mainly hydroxyl radicals. And these hydroxyl radicals then further damage other components of the cells like proteins, lipids, nucleic acids. At the same time, copper ions can also deplete sulfhydryl groups, and displace iron from iron-sulfur cluster proteins. So there are different mechanisms of copper toxicity, and it's not really known what is the main mode of action of copper toxicity. And what is very important also for copper toxicity is the direct contact of the cells with the surface. We are working together with material scientists from the University of Saarland, and they have developed a method, or they are working with a method called direct laser interference patterning, where they are using different laser beams focused on a surface to create different surface patterns. And by different laser setups, you can create different surface topographies. And this is the idea behind it is to mimic nature. So nature, there are naturally surfaces that are, due to their surface topography, antimicrobial. These are, for example, the wings of the cicada species here that have these nanopillars. And just by the surface tension of these nanopillars, they act antimicrobial. For example, here this was tested with E. coli. So this was the idea behind. Additionally, modifying the copper surfaces with this direct laser interference patterning to increase antimicrobial activity. So here you see E. coli on a surface that was patterned with direct laser interference patterning. This is a 3 micrometer patterned surface, so the periodicity here is 3 micrometers approximately. And it was shown that the 3 micrometer surfaces can reduce bacterial, the 3 micrometer patterned surfaces can increase the bacterial viability up to 15-fold. And this was for E. coli also. In the biofilms experiment now, so biofilms also has a very nice acronym here. It's biofilm inhibition on flight equipment and on board the ISS using microbiologically lethal metal surfaces. And the biofilms experiment studies the effect of reduced gravity conditions, so microgravity and Martian gravity, on the attachment and biofilm structure on inert surfaces. So in this case, we used steel surfaces as a reference surface, and the antimicrobial activity of copper and copper alloys with this direct laser interference patterning. The bacterial species we selected for this experiment, or they were selected long before my time when I started on this experiment. So it was, we have Actinobacter radioresistens, which is a very resistant bacterial species. So it's also very resistant to cleaning agents. It was isolated in a planetary protection campaign from Mars Odyssey orbiter. Then we have Staphylococcus capitis. This is the organism I worked in my PhD thesis on, and the strain we're using for the ISS experiment, for the biofilms experiment, is an isolate that comes from the ISS. So in a different experiment where different surfaces were exposed to the ISS indoor environment, this strain that we're using here was isolated. So it will refly, or it already reflow. And then the last one is Cupreavidus metallidurans, which was selected because of its heavy metal resistance and because it's frequently isolated from ISS drinking water. And all of these 3 species, they are known for their spaceflight relevance, but also for their biofilm formation. So they all form more or less strong biofilms and produce more or less biofilm matrix. Here I have illustrated the experimental sequence of the biofilms experiment. So I will go now step by step through the different stages we had for the experiment. And that started with the preparation of the bacterial cultures in the lab. So we took these 3 species and grew them in R2A medium, double concentrated R2A medium, and then washed the cultures and stored them in buffer, so in PBS in this case. So they were stored in buffer until the experiment then was activated on the ISS. This part our collaborators from the University of Saarland did, so they did the direct laser interference patterning of the different surfaces. So here you see steel as the reference surface, then we used pure copper and brass as an alloy of copper. And we used 3 different topographies. Smooth surfaces as a control, then 3 micrometer surfaces and surfaces with 800 nanometer of periodicity. So in total we ended up with 9 different surface types. Then this is the hardware of the biofilms experiment. It was built by Casa Italia, and the hardware can contain 5 of these surfaces. And for each container, we have 5 of the same type. So either we have 5 smooth steel surfaces or 5 patterned copper surfaces, and so on and so forth. So we always have 5 metal plates inside here. Then they are covered with a gas permeable membrane and a lid. And in between here is just an air pocket. And this membrane allows gas exchange. And then the hardware has 3 reservoirs. One reservoir for the bacteria, where they are stored, suspended in PBS. Another reservoir for the medium, which is in this case also double concentrated R2A medium. And then one reservoir for the fixative. And in this case, we use a fixative that is composed of PFA and paraformaldehyde and glutaraldehyde, which was specifically selected to contain the ultrastructures of the biofilms for later scanning electron microscopy. So here you see some images from how the hardware is assembled. So we filled the reservoirs, trying to avoid air bubbles. And then the experiment unit was integrated into the experiment containers that allowed automated activation and termination of the experiment. Then on board the station. So next step then was the launches to the ISS. In total for biofilms we had 3 different flights. The first one was in 2021, the second one 2022, and the third one in 23. We prepared all of our samples in the DLR at the DLR in Cologne in our laboratory, and then all the units were shipped to the US and launched. So we were just watching the launch through the live stream. And then on board the ISS, experiment containers were integrated into the cubic incubator. Here you can see Samantha Cristoforetti for the second flight of biofilms integrating our experiment into the cubic incubator. And the cubic incubator is an incubator and a centrifuge at the same time, so we can create different gravitational conditions. And for biofilms, we tested microgravity, Martian gravity, and Earth gravity as a control. And this is what it looks like. I think these are pictures from the first biofilms flight. So our microgravity samples, they're here on the outside positions in the corners of the 2 cubic incubators. So we used 2 cubic incubators on the ISS. And then here inside the centrifugal rotors, you have then the samples that received either 1G or Martian gravity. We had also our ground experiments at the DLR in Cologne, which looked like this, so a more simple setup, but did basically the same thing as the cubic incubator on the station. And this is now when we got our samples back, so it was maybe a bit unspectacular. This World Courier guy came with a cardboard box, so it was a bit hard to grasp that these samples were on the space station before. But you see everyone is here still wearing masks, still in COVID times, but still very excited that the samples were safely back in our laboratories. Then we disassembled the units, and I'm not sure if my video works here. Here you can see us disassembling the units. So for each flight we had 48 units, because we had 24 ground models and 24 flight models. And we worked here in a sequence. So the first of my colleagues was here opening the unit and retrieving the liquid, and then my second colleague here in the middle took out the metal plates, and then the third colleague here in the end disassembled all the rest of the units. And I think this is always the most exciting part when you then open the units, because you can directly, with the visible eye, see what happened inside the unit after the flight. So you can either see that a biofilm has formed or not, or the copper surfaces, they also, because copper also oxidizes, you can also see then the different colors of the aging material. So this is always, I would say, the most exciting moment, or was the most exciting moment. Then so in biofilms we have 3 different sample types. So first we have the metal plates, which are always 5 of the same kind for each unit. Then we have the gas permeable membrane and the liquid. And we're doing different things with these samples. So some of the analyses we are doing are, as I said before, scanning electron microscopy, but also EDX. For the membranes, we do light microscopy, and for the liquid, we measure the concentration of copper ions, release copper ions, iron ions, and zinc ions. We also do light microscopy and also absorbance. And I'm not going to show a lot of results today, so we are still in the analysis phase, but I'm going to show you some results of the scanning electron microscopy and also of the ICP-MS data. So here you can see scanning electron microscopy images from the biofilms experiment. And you can see here these upper two rows are steel and these lower two rows are copper. And then here you see the smooth surfaces, the 3 micrometer surfaces and the 800 nanometer surfaces. And then here you see the samples at 1G, micro G, 1G, micro G. And here I didn't include the Martian gravity in this picture. So now when we zoom in a bit first at the steel surfaces, so here again you see in the upper row you see 1G, in the lower row you see microgravity. You see that a lot of cells have attached to the surfaces. And yeah, by the way, I'm just presenting results for Staphylococcus capitis because this was also the species I focused in my PhD thesis on, so it's kind of my favorite of the 3. So I'm just showing the results for this species here. So you can see that the Staphylococcus cells have attached to the surface, that they have formed also small amounts of biofilm matrix. So you see here also these little appendices here. And sometimes you also see here very deflated cells. This you see a lot on some samples you see it more, on some samples you see it less, and we are not exactly sure if this is a natural phenomenon or if this occurred maybe like during transport or storage of the samples or later during the preparation for the SEM imaging. Yeah, but you can see technically here that there is no real difference between the 3 different surface topographies, and there's also no real difference between 1G and microgravity samples. However, I have to say with a disclaimer also that we are not finished with the whole image analysis, so we have a lot of scanning electron microscopy pictures, and we're currently working on like a semi-automatic way to analyze all of these images. But the problem is a bit that they look very diverse, because when we look now at the copper surfaces, it looks completely different. So on copper, we only see a few cells that have attached to the surface. So here again, we have 1G in the top row and microgravity in the bottom row. And what we also see is that the cells are a lot smaller than the cells that are attached to the steel surfaces. And you only see single cells that have attached, and you don't really see any formation of biofilm matrix. You see that some of the cells are covered that have like next to them these granular particles. So what we can say is that copper clearly inhibits the attachment of the cells to the surfaces and also the proliferation and then ultimately biofilm formation. So we didn't see any signs of a starting biofilm on the copper surfaces. However, we also didn't really, we didn't really see a difference between 1G and microgravity. But we also didn't really see a real difference between the 3 different surface topographies. So this is just sums up what I already said. So there is no real difference between the microgravity and the 1G samples, but we see that copper indeed inhibits biofilm formation, also in reduced gravity, but this was also expected. Now just shortly speaking about the ICP-MS data. So here you can see the concentration of copper ions, and here you see the ions released on the steel surfaces, which are, as expected, not a lot. And then you see here the ion release for copper and for brass. And what we clearly see is that the ion release on the copper surfaces is higher than on the brass surfaces. However, these data, this figure looks a bit chaotic, but it's also because the data are a bit chaotic, I would say, because it's very hard to find a trend here. So it's not really clear. So I don't see a trend. For example, I don't see that always cupraevidus metallidurans was the one that leached the most copper ions. Or I don't see a real difference that always in microgravity there were more copper ions released than in 1G. So it is really more dependent on the type of bacteria, but also the topography, how much ions were released. And it's not, so in my opinion, what I can say now, it's not so much dependent on the gravity condition. So I'm already at the end of my presentation, and I want to give a quick conclusion. So this experimental setup of biofilms is suitable for testing antimicrobial materials under spaceflight conditions, so it can also be implemented to use now, for example, different types of antimicrobial materials. What we saw is that the growth and the initial steps of biofilm formation and the matrix production is visible on the steel surfaces, but is not visible on the copper surfaces. So on copper, we only saw traces of damaged bacteria on the surfaces, and on brass, the same. Even though on brass, it's a bit of a different story, but maybe, you can, when the biofilms publication is finally out, you can read a bit more about it. So brass is a different story. Then the ICP-MS results, as I just said, they are very dependent on the bacterial species and the surface topography more than the gravity condition. And the growth and also the matrix production, it varies a lot in between samples. So this is why the analysis or the systematic analysis of all of the microscopic images we took is very difficult because the samples are so versatile. So the best thing would be to look at each image and count how many cells there are. But I think this would be a 10 years job for someone. We have actually a fourth flight of biofilms planned for next year, where we test different surfaces. As I said, we are currently still working on data analysis, but are also already preparing to put a publication out on the results. And with this, I want to acknowledge all of our collaboration partners. I also especially want to acknowledge Ralf Möller, which was the PI of this project, but sadly passed away this year. But he did a lot for this project. So I want to also give a shout out to him here. Thank you.
Katharina, let's hold the questions for the end, Katharina, and congratulations on your PhD defense. That you're now a doctor, Katharina.
Thank you. Thank you.
With that, I want to invite our next speaker, Jera, from the Universiteit of Brussels, and she's going to talk about the optical nanomotion detection in the microorganism environment.
Thank you for the opportunity to present to you our newly developed method, optical nanomotion detection, as a rapid antimicrobial susceptibility testing method for fighting microbial infections. So to start, as we already had a chance to hear multiple times over the last few days, astronauts face a number of challenges during space missions and in general being in a space environment. And one of those challenges is health problems. Reduced immune system suppression, reduced immune system coupled with containment vessel being contained in a space in microgravity where aerosols can just float around, having a lot of high touch surfaces, as well as increased stress. It all like lowers the immune system and the health risks of the astronauts. And when we add to that, on the other side, the high proliferation of microorganisms, as well as the increased virulence of the microorganisms and the increased resistance to the antimicrobials, it all leads to more severe infections that can also lead to sepsis. In these cases, it's very important to perform antimicrobial susceptibility tests to specifically target these pathogens with specific antimicrobials so that we can minimize the development of antimicrobial resistance, because this could lead to mission failures or The traditional methods of antimicrobial susceptibility testing, like phenotypic methods like broad dilution, disk diffusion, gradient diffusion, and others, they all take quite a long time to perform. If you don't think of just the incubation time, which is around 16 hours, just to perform these tests, it takes 24 to 48 hours, sometimes even longer with slow-growing strains. On the other side, we have genetic methods and mass spectrometry that takes less time, but these look just for specific resistant markers. So if we have a newly mutated strain, we don't know what to look for there. This is important because, as I said, to combat antimicrobial resistance, we need to have something that is very fast and that can be done maybe even in a span of a day. Here I will show you two clips of yeast cells. The first clip is yeast cells that are very happy in a nutrient-rich medium, and as you can see, they kind of move quite a lot. Whereas once we add ethanol to those cells, they completely stop moving. This we call cellular nanomotion, and we use it here as a signature of life. So basically, we observe and see if the cell is moving. We call it a live cell. If the cell is not moving, we see that it's a dead cell. How our experiments flow is we take a sample, we incubate it in here, it's a petri dish, but right now we are using microfluidic chips. We upload it onto a microscope. It's just a normal bright field microscope, make movies, and observe the cells. To be able to analyze these movies, we developed a specific software that cross-correlates first frame to the second, second to the third, so on, where we track the path of that cell. And with the alive cells, for example, at time 0 in growth media, this is real results here. We have a path that is quite large. You can see the cell moving quite a lot. Whereas a cell that is treated with 70% ethanol after, let's say, 1 hour, this data, yeah, it's after 1 hour, the movement of the cell is reduced quite a lot. To be able to perform antimicrobial susceptibility testing, we developed a microfluidic chip. This microfluidic chip is made of 3 layers. The first one is a commercially available EBD chip with engraved channels. Here it has 6 different channels. Then we have a layer of PDMS strips with microwells in it, and then the bottom layer, the last layer, is just a cover slip glass for support. Each PDMS layer is aligned with each channel in the EBD chip, so we have 6 different compartments, and we can perform 6 different experiments at once. So we can do like 6 different concentrations of one antimicrobial drug. Why we choose microwells is basically to trap the cells in microwells without the cell touching anything around it so that it can still freely move. The importance of the geometry of microwells is that if the sides of the microwell are the same or smaller than the depth, in that case the cell is going to be trapped inside, and when we have the flow over the cell in the channel, the cell will just circulate inside and never go out. That way we can easily exchange growth media or any nutrient-rich media with antifungals and antibiotics, and that way we can follow that exact same cell before treatment and after treatment over time. Here I have a little clip just to show you how the cell actually just rotates inside. It goes in and out of focus, but it never actually goes out of the microwell. So to be able to do this, we don't need any fancy equipment. Here, for example, we 3D printed a microscope, just a regular bright field microscope. The design is taken from open flexural delta stage and just slightly adapted to our project. As you can see, the imaging is quite all right, so we just need a little camera, a microscope, and that's it. Here it's a preliminary stage of our analysis where we can, before tracking the cells, so before doing anything to the movie, we can just look at how much the pixels change. So in red are the pixels that change the most, and in blue are the pixels that change the least. That way we can see if we have anything moving or if it's a clip worth analyzing or not. So now I'm gonna show you some data. Here are some nanomotion of different bacteria. For this experiment, we incubated E. coli cells with different concentrations of ampicillin for 2 hours. So here we have the control, 0, so with 0 ampicillin at time 0, 0 micrograms per ml of ampicillin at time 2 hours. And we can see that there is a lot more going on, a lot more of the pixels changing. Then we have 10 micrograms, 100, and 250. We have a sensitive strain and a resistant strain. So if we, for example, compare 2 hours of 550, here we see that there is still a lot of cells. They quite proliferated from the time 0. Whereas the sensitive strain has very few that are still moving. We can present our data also as the displacement per frame, and here we see that the resistance strain stays, there is no significant difference between the concentrations after 2 hours, whereas with the sensitive, the drop in the nanomotion is quite significant, both for 10 micrograms, 100 and 250. Same was true for Staphylococcus aureus, treated with vancomycin. We have 1, 10, and 50 micrograms per ml, and then at the end, the 50 micrograms per ml were killed with UV additionally, and we see also a slight drop from here. That means that possibly here, 50 after 2 hours, they weren't completely all of them dead. Then after we killed them with UV, they were all dead. E. coli with doxycycline. And here, the graph B is a very interesting one. It's Mycobacterium smegmatis, which is a model organism for Mycobacterium tuberculosis. With standard methods for this bacteria, it would take us weeks to perform antimicrobial susceptibility testing, whereas here, in 2 hours, we already see a drop in nanomotion for 1, 10, and 50 micrograms each. So, on the other side, we have performed also the optical nanomotion detection on yeast cells. Here we have 3 different strains of Candida albicans. In the first group here, we can see the Candida albicans DSY294 treated with 3 different antifungals, and we can see how this specific strain reacts to 3 different antifungals. So we would say that amphotericin B here, the drop is quite fast. After 1 hour of treatment, it drops quite significantly, whereas for caspofungin and fluconazole, the drop is more gradual. To be fair, fluconazole is a fungistatic, not a fungicidal, and we did afterwards do some experiments that I didn't show the data here. So after we reintroduce, after, for example, 5 hours of fluconazole, we reintroduce YPD, we see again a peak in a nanomotion, so the nanomotion goes up again. Here we have a candid. Here we have DSY 1024 and 4614. One is candin resistant, one is susceptible, and we can see the difference. With the resistant one, the nanomotion doesn't go down, whereas with the susceptible one, the nanomotion goes down. With this technique, it's quite an interesting thing that we can follow also single cells over time, which is represented here. So for A, we have single cells, 20 single cells represented. So the cell 1 is the same cell at time 0, at time 60, at 2 hours. And we can follow the nanomotion over time. And we can, for example, here with the treatment with caspofungin, actually see that there is quite a difference in responses between single cells and the population, whereas, for example, cell 20 didn't respond quite well to 0.5 micrograms of caspofungin, etc. So we can maybe see in a bigger population quite fast if some cells are more resistant or more tolerant to a drug or not. And finally, I'm gonna show you also this way just to represent it. So we have the total displacement per movie, per time point of a single cell, and we can see that actually it's not a straight down, it can go up and down. But yeah, it's a nice way to represent single cell data. So finally, I'm gonna conclude with the potential of optical nanomotion to transform space and Earth-based medicine, because even here on Earth, it could be a good diagnostical tool that could be included in every hospital. It's rapid, it's label-free, it's very useful for targeted treatments, and it's nice that you don't need any fancy equipment to do it. We will evaluate this method using a specifically developed microfluidic chip for the Flumias microscope to test this method in real microgravity. We hope that it could become a standard diagnostic tool for both long but also short space missions, and potentially also as a method for detection of life, because it is a signature of life in the end. With this, I would like to thank my PI, Professor Ronnie Willard, as well as my lab, our collaborators, and you for your attention.
Thank you very much. We keep the questions for the end. I think there will be a lot of questions. I already saw the hands, but we have a lot of time. Don't worry. I would like to invite the next speaker, Natalie. She is actually going to have 2 talks today, and I think you enjoyed actually 3 projects, right? You're going to talk about it.
2.
Floor is yours.
Okay. So good morning, everyone. I'm Nathalie Les. I'm working at SCKCN, which is the Belgian Nuclear Research Center. The research in our team is anything which deals with biotechnology, microbiology, radiation, radionuclides. But today I will present our ESA-HRE C research or experiment, which is a photobioreactor with cyanobacteria for oxygen production, as well as, we hope, edible biomass for life support systems. So maybe during the talk I will use the word Artemis. It's because 20 years ago the proposal was called Artemis. In the meantime, that became a name for other missions, and we changed our op name to ESA-HRE, but in my head I'm still working on Artemis. It's actually a project where we try to develop a photobioreactor into a very small scale one that can run almost automated in ISS, with the idea of testing how a cyanobacteria is actually kinetic-wise possible to produce oxygen and to grow, also to produce biomass. It's part of our contribution to the MELISA program. MELISA is an ESA initiative where we look at future life support systems with the aim of course, producing food, water, oxygen, but by using some of the waste to do this. The cyanobacterial reactor is part of this loop, which aims to provide oxygen to the crew, remove the CO2, and also to provide some additional edible biomass. We use spirulina in this bioreactor, one specific strain. In the meantime, it's changed also names. So the Arthrospira C experiment was the old name. Now it's called Limnospira. But okay, it's still the same organism. We have in Melissa community a pilot plant in Barcelona where we already test this strain in larger scale. The crew in this case are rats, which are living off the oxygen which is produced, which we tune with the lights to have more or less. The aim of the flight experiment is just to see how can we do this in radiation-resistant space biotechnology that fits a space station. Still, our experiment is just a proof of concept. It's not in productivity anything related to what we have in the pilot plant. We had a first experiment in 2017. We realized that space biotechnology, from my perspective, is not well developed. First of all, there is the whole issue of how do you get the whole active bioprocess in dormancy, then get it up to space and reactivate it again with the same efficiency. How do you do this in biocompatible, sterile ways? The aim of Arthrospira C is to have a full actinic reactor, so one strain only for the full duration of its run. The gravity-independent operation of such a bioreactor in fluid dynamics, the mixing, the gas-liquid transfer, because we are actively producing oxygen. Operator independent, so we work with timelines, programmed timelines of feeding, of measurement, of dilutions. So this based on predictive modeling, based on the temperature, the light settings that we have. And then we try the online monitoring to see if what happens in space is equivalent with the parallel ground experiments and also have the options to command. There are some papers of this flight experiment. There is also an overview, a review of what other experiments similar like us have been doing in space. Still, I think the online monitoring and the kinetic follow-up of bioprocess is really a challenge. I also want to say the biology that you choose for bioreactors, it's spirulina. You think of a spiral organism, but in fact, over the years, we worked with this organism. We selected what I call a lab-adapted strain, maybe a space-adapted strain, because we wanted to be robust to this dormancy, to come up again when we give it light. We don't want it to produce a lot of EPS because then it's sticking all over in our liquid tubes or on our membranes. We don't, so spirulina has these gas vesicles to float or sediment, so we have strains which do not float anymore because it's easier to have them in suspension. So a lot of tests have been done describing this strain, what is the best dormancy revival conditions, how do we propagate it again after this dormancy phase. We did simulation of irradiation, impact simulation of microgravity. I will not present all, I just have a few slides on this radiation simulation, microgravity simulation. We also, in the meantime, developed a cryopreservation method for this strain and other spirulina strains. So there's a lot on the biology that you choose. It's just not any strain. Irradiation, we just use facilities we have at SEK. Is it an ideal space radiation simulation? No, but we do the best we can. We aim for long duration exposures, 8 weeks. We were able to do that in what we call the LNK calibration rooms. We use cobalt-60 here. We tried here to go to a dose rate which is a bit more than ISS to at least see how stable, how well it performs. The unique thing is that we try it to do with active growing cells in photosynthetic state, continuous elimination. what we found is that there is a bit of what we call hormesis effect, that after 4 weeks of these batch cultures, there is an increase in biomass productivity in the irradiated cultures. We did 2 types of these experiments, one where we go from very low or lower cell densities to higher cell densities. It means that also the light flux to your culture changes. It has a bit of more sensitivity to the radiation. If you keep your cell density constant, your light flux constant, it has less sensitivity to the radiation. But the response was also intermediate. It also goes off again after 4 weeks, but the main message is it didn't care that much about radiation, so that's a good sign for this strain. We also did simulated microgravity experiments, which we use this RPM and rotating wall vessel setups to short. If you don't mix in this culture, the oxygen gets stuck, it doesn't diffuse, or there is of course this resistance for good diffusion, which hampers the fixation of CO2 at your Rubisco sites and enzymes. There is competition between CO2 and oxygen, which has a whole cascade on some of the metabolism. This is what we don't want, because in Melissa we want active oxygen production, which we can control and steer, meaning that we confirmed here that in our bioreactors we want full mixing, convective mixing, and we don't want to have the impact of microgravity. So the bioreactor design for space was done by Redwire for ESA. It's a small biolab compatible hardware. It was quite a challenge to get all of what you normally have on a bioreactor in. We don't have all, so we are partially running still blind. We don't have pH sensors, no dissolved oxygen sensors, no CO2 sensors, no liquid pressure sensors, but okay, we tried with the model to predict and go around that. Inside it looks like that. It's a very small, or I think still a very small culture chamber, 50 ml. With a polycarbonate window, we have a LED array. Inside we have this white gas permeable membrane. The gas chamber is at the other side of that membrane. On top of the membrane we have a stirrer, but to hold a magnetic stirrer bar, but to hold that stirrer during launch vibration, there is this brass bracket and a little cup to hold the stirrer bar. Our aim was to keep all the time bubble-free, perfect mixing, with the aim of have homogeneous elimination, good gas transfer, but also biomass measurement. So we have liquid loops going over a pump, but also going over optical measurement unit, where we measure optical density and fluorescence. And then you have on top these feed reservoirs, sample reservoirs, which we plug in here. And then all of that is controlled. On top of these feed reservoirs, we have some radiation sensors. We are a nuclear institute, so we send in everything we have, also radiation sensors, because we have a lot of metal. This metal box goes in biolab, which is also a metal box. So it's quite different radiation doses that you may get inside this than outside. So this was the 2017 experiment. I don't know if the movie works. Yeah, at that time we had to work in the glove box of biolab, and that's how the reactors were installed on biolab. Did it work? Yes, it worked. We had actually a model predicting what we could expect. It's not my expertise, it's from our partners in the University Clermont-Ferrand in France, which is a Melissa partner. It includes several parts, so it's really a photobioreactor we run on light control. So it means at all times the cells are starved from light, but it means also that if we increase light, we can increase oxygen production. So that's our control parameter. Because we have in Artemis B the batch condition, there was also a prediction of the pH increase due to the bicarbonate. I forgot to say, we did not have the capability of having CO2 from the atmosphere of the station getting into our bioreactor, so we are providing the CO2 as bicarbonate. We have also substrate limitation due to the batch condition, and we have this gas membrane, and we are actually monitoring oxygen production through pressure, and that was also part of the model. What we were expecting theoretically in perfectly mixed conditions were these values. In the paper you see very different values, because some of the batches we had worked okay, some others did not, and based on the modeling we predict steering issues. Some of the hardware came back, and we indeed saw that post-flight some of the stirrers were blocked, and probably we were not having good mixing. That led us, okay, so overall we were just happy that we were able to do the bioprocess. We did keep a good point to say, we run for 1 month on board the station with multiple crew interactions, but all these interactions we were maintaining the full axonicity. It's also how we designed all operations. That was a nice achievement. But then we made a step up to Artemis C, which was the aim no longer just to do some batches, but to have a continuous operating reactor. Also not just one light setting, but that we prove that we can control the production by changing the light setting. And that's the whole concept of Artemis C. There were some improvements on the mixing. We had to have a way of this feeding, continuous feeding, but allowing also measurements, dynamic control by light, and we also went for a longer exposure, 9 weeks. So that's, yeah, maybe a message here is that during all these tests, we learned that mixing is a key in our biotechnology. We were really seeing biology-wise that it was not mixing. While PD said, no, all the dye tests that we did shows that we have perfect mixing in this setup, in this design. We pushed until we had additional people doing CFD modeling, and there came that indeed we have high fluxes close to the stirrer, but all the blue is actually dead zones because of these additions that were made to keep the stirrer bar in place. Of course, all of this came late, and design cannot be changed anymore, but then, yeah, we found solutions, workarounds to have stirrers turning at time like this. Then it has a ramp up, a ramp down, it turns back to still try to improve. Also, the similar thing goes for ground hardware. So this is an example of how we run these things on ground. We initially were running a worst case scenario where the gas part is on the bottom, the liquid on top. So if we were able to get gas from liquid to the gas chamber at the bottom against gravity, we were happy, but we didn't think that all the liquid loop where we are doing our measurements is also, and it's not mixed, is also exposed to gravity, and we were not measuring what we were having in the reactor. So we designed this anti-settling device that helped that we were also measuring the right things in the liquid loop. And my main message for scientists here is please push for correct multi-phase gas liquid, liquid solid mixing in all your setups. I know there is a lot on microfluidics, but also there, I think CFD modeling from the start in your ships, in your design, has to be on board and not later on, because then you cannot change your design. And it's key for all processes you see on your cells. So yeah, Artemis C, we run through ESR, PDR, CDR, MRR, hardware testing, science model testing. We did science verification tests 2 times because we found a lot of issues. We did experiment sequence tests. So I will tell a little bit on that. It's also not all positive, but okay, there are positive things. So science verification tests is testing your hardware for the interaction with your biology. We do it on ground models. It's not the actual flight hardware, but with software we measure pressure buildup in different light conditions, 45 micro-Einstein 50. We test how much oxygen we detect and produce. That worked okay. What happened is, although that we have runs where it worked okay, in other reactors it didn't work. We saw biofilms, we saw liquid going out that we didn't expect. This is all post-run inspections. So there were loss of liquid pressure, there were quite some variability between the reactors. So we still had a lot of the parameters and the run settings to optimize, also the way we release the oxygen. So in initial breadboard models, we were actually flushing the gas chamber with nitrogen gas. In ISS that was not possible, so now in the gas chamber we have 100% oxygen, which of course has an impact on the biology, and we have to also change our run parameters. We also use these tests to test some of our omics analysis that we prepared on these samples, so that also worked. Yeah, you can find all this in some of the publications. We did other tests, we call it delta SVTs. We improved, still one was different from the other, so you find solutions to deal with it. Then we do EST tests there, EST experiments sequence test. I think typically are not done with biology, but we push to have biology because the only moment where biology can be really in contact with your flight models. And we saw that detoxing, sterilization of the flight models and then testing how biology interacts is key, because you don't want to put in your biology in something which smells like a new car and then say, okay, you go to space and now you do it. So flight hardware should be run in contact with biology before it goes to space, which is not the same idea of ESA all the time, I think. Again, during EST, a lot things worked well, but 2 of the 4 reactors didn't get feed. What happened is that it's new hardware, new septa on the reservoir, new needles, and some of the needles, by puncturing these new septa, got blocked. Gas chromatography people doing autosampler and companies know this very well, that you should use needles where the hole is at the side. PD didn't know it. We never asked what kind of needle was on it, but then all of a sudden, if you start using new septa, you discover it. No design change possible, but okay, there is polishing on the needles. You reuse the same septa because then your risk of these events is smaller, and so you find workarounds. But every test counts. So where are we now? We are here today. We actually launched 4th of November, Artemis C from Kennedy Space. We did all the preparation. Hallelujah, they woke up after the upload. After 1 week, we had this propagation. Because in Artemis B, some of the biology didn't wake up when it was arriving at the station. So, but we improved our inoculum preparations. We have now one cycle, 2 weeks of production phase. Some of the things, so we prepared 60 of these feeding reservoirs to cover 9 weeks of operations in space. It's quite busy, but you've seen all these pictures, I think, from all the scientists preparing samples and things. It's a quite stressful period, but if you have enough team and redundancy, if someone gets sick, you get it done. So please prepare for backups. Also, what we learned is that these bioreactors, they are programmed, it's technology, it's electronics. So we, as scientists, biology in labs, working with these bracelets to counteract static electricity. Then the engineers, we learned what to touch, what not to touch with gloves, how to disinfect. Then the programming needs to be done, the interactions, the safety people. It's a learning process of years, and we were happy we're the same people from the same company for the last 5 years, because there is a lot of back and forward training between engineers, biologists, what is partially of the sterility that you can do, you cannot do, but it worked. And I was very happy to do the handover on the 3rd of November, and then even 5 days later I saw the same box floating with the astronaut, which for me was a very nice achievement after many years of work. These are the data of last evening of one of the flight models. So yes, we have still oxygen production. The other three are looking like this. One is not looking like that. We have much slower oxygen production. We are still figuring out what is going on and what could be done to maybe rescue or remediate it by commanding. Why are we doing this? With the aim of having the step up to larger reactors. So in the Melissa team, there is other people working on a project called BioRat, where we will actually do active air revitalization. So we use the CO2 of the cabin and release the oxygen. My personal dream is that maybe once we can also do it fully remotely on the moon. We propose that, we will see if it ever happens. But of course the final goal is that this is kind of biotechnology that can be used in a life support system. So all this goes back in what we call the Melissa community, and learn from it and use it maybe even for other algae setups or other configurations. There is too many people to thank, but there is of course all the sponsors, all the scientists, all the community, but it's many people, all the ESA teams, because the current running experiment for 9 weeks for operations, it's really a challenge. So the MUSC teams at DLR, at Biotesks are doing their utmost best to help and support it. So that's it. Yeah.
Thank you. We keep the question for the end, but
Okay.
because you are replacing Felice Matuleo
Yes.
because you are co-I in this project, Urinus,
Yeah.
and you please continue the presentation with the next presentation, please.
Okay.
And then after all, we invited the speakers to come, and we can have a discussion. If you can keep it a little bit shorter for the time's sake.
Sorry. So the next project I'm presenting is actually led by Professor Ramon Ganinet from the University of Ghent, but we at SEK are co-I, and it's testing how we can use synthetic microbial communities to convert urea from urine to nitrate for fertilizers. Again, it's linked to Melissa, and I will skip all the information, but this time it's on this compartment in the Melissa concept, which does the urine conversion to nitrate for using that as fertilizers for plants. The bioreactor or the bioprocess that we are using is actually a consortia, because these reactions from urea to ammonium nitrate is not possible by one bacteria. It's a consortium. In nature, it's very complex, dynamic consortia. We went for a gnotobiotic or a synthetic community for strains which can do each of the steps, but we also know work together. So there is the ureolysis done by Comamonas. There is the nitrification step in 2 steps with the nitrophyres, Nitrosomonas, Nitrobacter. The question we wanted to answer is how are these bacteria and their interaction influenced by space, because single organisms is one thing, communities is another thing, and I think we still lack some information how communities work in space. The idea of Urinis experiment is indeed a kind of a flight experiment to ISS. We prepare everything. We have a batch run of this conversion, this process. We don't monitor it. It's a one-shot batch kind of process. We bring samples back and we analyze it. It's scheduled now, I hear, for 2027. But in the meantime, we are preparing that. We are testing microgravity ionizing radiation impact on all of the bacteria single, but also in what we call a tripartite community. We use a synthetic urine-like growth medium, and we look, is ureolysis still ongoing, is nitrification affected, but it's endpoint measurements, it's not kinetic measurements. The method we used at SEK is whole transcriptome analysis. Some other partners are doing the proteome analysis and metabolomics on the same samples. The simulation of microgravity we use is, again, this rotating wall vessel and random positioning machines. We did 3D print different things to do it in culture bags, because for this process, oxygen, again, is very important, but this time oxygen to get into the cultures, because it's the oxygen is the electron acceptor of the conversions, and it's part of this redox couple that you want for the process. So oxygen supply is very important. What do we look at? What did we detect? Genes being downregulated or overexpressed in this simulated microgravity. The details are, because there's a lot of gene lists, are in the paper. They're also deposited in public repositories. What we see is that it doesn't happen that easy in simulated microgravity. The growth is slower. The robisco, so the nitrifiers are autotrophic, so they are fixing the CO2, is also slower. We do see that there is motility going up, biofilm formation going up, or denitrification going up. We think this is again a starvation-related response to the fact that oxygen and CO2 is not well going in and out if you have this low shear environment and not well mixed. But what was good to see is that actually the kinetics, or not the kinetics, but the expression of the genes that we need for ureolysis and nitrification were not affected and were still continuing. The denitrification was a bit of a worry. So we want nitrification, ammonium to nitrate, but if you don't have sufficient oxygen, it means that the nitrate is used again as electron acceptor, and you go through another process and you end up with nitrogen gas. It's interesting. There is another project in Melissa actually doing this to replenish the atmosphere in cabins with nitrogen gas. But if your process and target is to keep nitrate as fertilizer, you don't want that, and you actually lose part of your nitrogen, which means your process loses efficacy or efficiency. So yeah, we don't want denitrification in space, which means that you need to take this on board, because you need active mixing or methods to get high oxygen supply to your cells. We did ionizing radiations. First of all, we checked a bit the radiation sensitivity of these strains. We found out that nitrifiers are actually quite sensitive to radiation, more than an E. coli. But okay, you still can go to 400 grays before you have impact. It's not doses you would expect in ISS, but it's something to keep on board, that these processes are not so radiation resistant. Then we did also simulation with neutron radiation, because some of these tests are again with gamma, but for having some test of the high LET effect, we did in using again some facilities at SEK neutron irradiation. It's again a shortened lifetime test, because we gave a dose of 4 months in a 3-day period, but cost of radiation rooms is high, and so sometimes we have to find the best setup practically. What do we see? We do see some response of the bacteria in several metabolic pathways. Of course, what you do expect is that iron, you don't want iron because it creates ROS. The sulfate groups and the thiol groups, you'd see some shifts in metabolism there, DNA repair, the chaperones, protein repair, I think the normal systems you would expect. Of course, what is important is to know if you tested actinic in single or in tripartite, how does it respond different? We do see that there is another response sometimes when they are in communities. And that's, I think, also a take-home message, that if you have bioprocesses with multiple strains, you also have to figure out how they react together to a certain stressor. But overall, I think the message we saw is that the community itself is quite stable. The composition, because you need all 3 of them in a certain ratio, and they stay stable, which is again a good sign. Yeah, that's the conclusions. The general outcome of our study so far is that it seems microgravity and radiation don't impact too much the process steps we want, but we have to take into account that efficiency may go down due to some denitrification. Of course, this is all just based on simulation and transcriptomics. There is more work to do. There is the proteomics, the metabolomics. We didn't study the biofilm structures because these compositions or these strains like to be in biofilms. We only did endpoint analysis, nothing on the kinetics, so we also don't know that. But again, we are preparing for a flight. We are just gathering all the ground-based work to do as good as we can the flight experiment, which is coming up. Voilà, this was Urinis project. Yeah.
Thank you very much. So I invite all the speakers to come and have a seat here when you can have a nice discussion. And first of all, I will open for the audience, because I saw that some people were already ready for a question. So any question for the audience? Yeah.
Jean-Pierre. Yeah, thank you for all these very interesting talks. Particularly, I've a number of questions, but I try to focus particularly to Vieira. You have mentioned this motility work, and I have some, I would say, understanding problems maybe. You mentioned yeasts and Candida albicans, this is right. And what kind of motility is it? Because you say nanoscale, is there apparatus in these organisms?
So we do it for both motile and no motile. It's not related to their mortality in general. It's just that the cell on itself, we don't know why. We're not sure, but we assume it's something to do with the metabolism of the cell, that it randomly moves, and the movements are in a nanoscale.
Did you have checked also the physics? Because this is depending also on the medium, particularly on the viscosity, on the temperature, on the density, which also causes a motility without in any active motility of the cell. Therefore, this is something you have to take into account. Also, because you said also this is a clear biosignature. In this case, I would say no, because I know about fungi and also yeast and so on. They have no apparatus and they are not actively motile. So in case of bacteria, this is another question. But in this case, this is critical. So just check it. Check also the other possibilities for this motility, because according to the media you're using, there could be these differences you have shown.
Yeah, but we follow, for example, the same. We do it for both yeast and bacteria, but I'm more comfortable now to speak about yeast.
This is my question. Yeast.
So we follow it in the same media, so it's the same cell, and we kill it, for example, with UV, and it's in the same media still, and it first moves, and then after we kill it, it stops moving.
Yeah.
And it's in the same environment. The temperature is stable. We control the temperature.
Then also
we
check.
don't have convection. We checked for that as well.
But even this can be also explained by physical, because you are changing also radiation, sometimes electrostatic reactions. So just check it. Otherwise, you could have some troubles also if you want to publish it, because if the physics are looking there, there are other explanations, branch movement, electrostatic density, and so on. Just check it.
Okay,
please.
thank you.
Thank you, Jean-Pierre. Any more questions from the audience? Yeah, David.
Thanks, Rodrigo. Hi, congratulations on everyone's successful work. It's really inspiring to see your stuff on the ISS and doing so well. Katarina, I had a question for you. Mainly, I was curious about maybe if you could just share your experience of your hardware assembly, particularly in regards to how you maintained sterility of your samples. For me personally, that's something I'm super paranoid about. So any insights you have would be really appreciated. Thanks.
Yeah, I think we use a lot of bacillol. So I think in the end of the day, we were all a bit like, whew, from all the fumes. No, so yeah, what I was most afraid of also was contaminations. And actually we had, I think in total we had 144 containers, and we had in 2 containers we had contaminations also. So I think it's a good, it's good, but still not perfect. But we worked under a sterile bench. We autoclaved everything that was autoclavable. But for example, the copper plates you could also not autoclave. So there we use UV sterilization. And then, for example, we had all of our tools. We had them in ethanol before we put them on. Before we screwed the hardware together, and we put them back in ethanol. Frequently exchanged the ethanol. But I think we were lucky that most of our hardware components were autoclavable. So, and then it's, when you have them all in separate bags, I think it's then easy. And for the bacterial cultures, when I prepared them, I prepared 3 cultures, and I checked all of them for contamination before then actually choosing one of them. So 3 cultures for each strain.
As a follow-up for that, but maybe Natalie, you can add something from your experience with your hardware for a biolab.
Yes, sterility, a big challenge. Over the years, we learned that to assemble the maximum you can before it goes into the autoclave, so that the handlings after it comes out is the minimum. So we had this bioreactor with all these tubes hanging out with clamps and everything, but then only the operations was to fit it of the tubings into the peristaltic pumps or the valves, but to assemble as most as you can before, so that the operations afterwards. We are very scared of using organic disinfectants. We don't want that. So we use a lot of hydrogen peroxide because it doesn't leave organic traces. What we also know is that autoclaving is also leaving some traces of gassing things. So we have procedures, we call it our detox time, is where we fill the whole hardware with medium that we plant, and it's important that you don't do it with water, but with the actual medium, with the pH and everything you want. We let it sit for weeks, so that it dissolves all what comes out. We remove the liquid. We test that liquid for toxicity by we inoculate it. And we redo it if we have to, because we did test all the toxicity of all the components separate, but in an assembled hardware you bring all the toxicity together and you still have an accumulative effect sometimes. So yeah, we do a detox period for the hardware. It's also lessons learned we had.
Yeah, very good. Any more questions for the audience? Yeah, Jean-Pierre.
To Natalie. I'm very happy that you mentioned also that it is necessary to do a lot of work before, particularly also in the ground studies, because you mentioned also the SVT and EST and all this, because often at ESA we have this problem, they say, oh, you have to reduce and so, but with that we find out what can be wrong and then you have not good results, and this is really necessary. I would like also to support you. And by the way, you have a cool t-shirt. At Musk, we will be also looking on this t-shirt. We have to talk about it.
Very good. We can share the design, absolutely. Yeah.
Any other follow-up questions? Yeah, Rosa.
Thank you. So these talks were all amazing, so thank you. Maybe a follow-up question for Viera is so you mentioned that that all maybe hopefully will convince us. So you mentioned that you check and you are sure that the microbes were killed. So how did you check? Did you have like a colony forming unit, or I don't know, another way of checking that when they were stopped moving they were actually dead?
We did staining.
Ah, okay, like dead and live.
Yeah.
Okay.
So, yeah.
Okay, thank you.
Yep. Another question here from Boris.
So thank you very much for all your presentation. I have a question for the antimicrobial material. In your space experiment, you test the adaptation for 2 weeks, but for long-term space flight, Moon or Mars, it will be really more longer. Do you investigate the potentiality of evolution and selective pressure to maybe at a certain point, even if at the beginning it works well, maybe you will have a selection pressure and finally a biofilm. So do you have in your laboratory a whole sample that was looked at different time for maybe years, PhD time, for example, to see if finally something emerge in these conditions? Thank you.
Yeah, actually in that setup, no. I think that would have been a good idea. Maybe I will start it. Yeah, but we are looking in the lab. We are looking at copper, like adaptation to copper stress, because it's especially also in bacterial species that already have antimicrobial resistances. I think it's also you also sometimes then have strains that are additionally copper resistant, and this is, I think, I think this could definitely be a problem when using copper surfaces excessively as antimicrobial surfaces, that you would, like you see with antimicrobials, that you see antibiotics, that you then see resistances. But in this biofilm setup, we actually haven't followed up on that. No, I think it would be a good approach.
Maybe it's a good time for you to tell about the next mission, because you were kind of let's say selected for the ready to flight, because it was just there, and maybe you can, because you didn't comment on your slide, maybe you can comment a little bit about the different materials and
I think that and we are also only testing there 2 weeks, but we are testing different surfaces that have a different patterning. That is more similar to these nanopillars that are on the cicada wings, to then add the surface tension also to the structure, and not only have the chemical component of the copper. Yeah.
Great. Any more questions? What is?
Yes, sorry. And okay, if it is, let's say, toxic for bacteria, did you test also for human? Because if we are starting to put copper everywhere on board the gateway on something else, did you check for all reactor? That might be interesting.
Yeah, we didn't check it, but there are publications about it. So I think for humans, I don't want to drop now any numbers, but I think it's like 20 grams or something of copper that you have, or 20 milligrams. I'm not sure, but it's a lot of copper that you have to ingest before it actually is toxic. And so copper surfaces, there would be more applied, for example, in touched surfaces. So when you come into contact with your skin with copper surfaces, I think there is so little copper that would even reach your system that it would become toxic. Yeah, so I think these are just different, a question of concentration. Yeah.
Any more questions for the audience? We still have time.
Yeah, hi. I have a question for Vera as well. So in your data, the kinetics of the motility, basically, can you also correlate that to the classical analysis methods of like resistance tests? Like when you say, okay, like 50% motility is actually 50% viability, or like, can you define a minimal bactericidal concentration or anything from that? Can you derive that from the data?
So yes, we did MICs. We did MIC testing, and we were able to do it from the data that we obtained. The problem is just we have to standardize for how long we have to do optical nanomotion detection, because 2 hours, for example, the data that I showed here for bacteria, I think it's too short. So from that data specifically, we should reach a minimal plateau to be sure that that's when the cells are dead. So for use, for example, we are doing now maybe 5 hours of the data just to, for example, do MIC. But what we are hoping to do with enough data is to also look at the slope. So when we see the slope getting more negative irreversibly, then we can say that the antimicrobial has an effect or not. So we don't maybe need to wait for the cell to be fully dead, but yeah, we need a lot more data for that, and that could be done like in an hour or so. There is a company, Resistel, they don't do optical nanomotion detection, they do just nanomotion detection. So they apply bacterial cells on a AFM, atomic force microscope cantilever, and they detect the oscillation of the cantilever with the same machine, so with the detector, with the laser, and they're right now doing clinical trials, etc. So they are, I think, doing with bacteria around 2 hours, still not waiting for them to be dead, but just looking at the slope.
And also, let's get a follow-up. So we saw that even when they are dead, there's some residual signal still left.
Yes.
Do you have do you have to define a threshold for every single organism where you say like, okay, now we say they're dead basically, or is it all the same?
We would probably need to define a threshold for groups of organisms.
Okay.
I apologize if I missed a lot of the discussion, so maybe you discussed it already. My question is to Katharina. It's just a practicability question, more or less, because from the kitchen perspective, you would not choose anything with copper because you probably would just erase all the structure and also the copper after the TENS cleaning, whatever. So for long-term space flight, what is your recommendation? Would it be better to use some kind of copper material to make it more antimicrobial, or would you see it from the more practical point of view, and for cleaning it's probably much better to use steel or whatever, or aluminium? You cannot just clean it with some alcohol and that's pretty much okay for the next couple of days. So would you recommend to have a certain amount of acceptability for biofilms or for some kind of material also to change the immune system? Because you don't want to have a fully cleaned room and environment, because then you have a weak immune system. And when there's a new astronaut coming, then you're probably completely unprotected for any kind of pathogens that comes with a new astronaut. So that is all a complex situation, of course, but what would be your recommendation?
To be honest, I'm not sure, because I think I have the same opinion as you also, that it would not make sense to have everything completely sterile. Also, it's like it also doesn't make sense. But I think it's especially for critical surfaces, or also surfaces that are hard to clean. So where you can, for example, inside pipes, or that where you, for example, then use a combination of methods. You use copper, but then at the same time you use also regular flushing with disinfectant, for example. So I think this needs to be established in the future, like to find a good balance between antimicrobial but also not completely sterile, because that doesn't make sense. And it's also not feasible. You will never achieve that.
One more question, and then we have to end the session. Yeah, Adrienne.
Thank you guys for the great talks, and Natalie, definitely want to talk to you afterwards about the mixing, how you did that. I'm curious for you as well. Now, I mean, I really respect that you were able to do a programmatic, step-by-step and iterative approach to the bioreactor, be able to, at the next flight, improve things from the previous flight. So what is the projection? I saw that you're wanting to have a proposal for the moon, but in the intermediary period between end of ISS and getting to the moon, how are you going to keep that progression going until in the intermediary time?
Yeah, so Arthrospira C will stop, our project will stop after this flight, but all the know-how that we gathered is actually distributed in the Melissa community into what we call the BioRat projects, which are more tech demonstrators. So the volume of the bioreactor will be increased until 1 liter, 1.5 liters with all the fluids around. It's another team, it's another hardware company developing it, but we are removing from having a membrane on top of the liquid chamber. It's a gas exchange unit separate. There is a harvesting unit for the biology separate. There is different elimination systems, but the principles of mixing, of gas-liquid separation, liquid-solid separation, these kind of lessons learned, we try to bring into the other experiments. And it's multi-people involved, but not anymore for me, I think. But I really hope that all the lessons learned can find their way. And maybe not even only in Melissa, but in other if we do food fermentation or I don't know, organs on a chip, where there is also CO2 to go to the organ and oxygen, or CO2 out and oxygen in, and things like that. So.
Well, I hope you're in touch with ESA, whoever the intern, the lucky intern that's chosen for this life sciences handbook and the lessons learned. Make sure to get in contact with them and get that, because it'll be so valuable for the whole community.
Okay.
Okay, thank you very much for this session, and I hope to see you later. Thank you very much. Big shout.